So, so whereas the immunoglobulin therapy that we just talked about is passive immunity, okay, is specifically artificial passive immunity. Vaccines, on the other hand, vaccine-induced immunity is known as artificial active immunity because they induce the adaptive immune response. And the vaccines induce the adaptive immune response via the humoral response. So the humoral response, which is via antibodies to kill the free viruses, so the virus is not in cells, and then the cellular response, which is via the T cells to kill infected cells, okay, where the virus is in the cell. So in this slide, we'll talk a little bit about the humoral response, but this will be brief as you will be discussing this in much more detail in immunology next term. But briefly, humoral immunity is also called antibody-mediated immunity. And with assistance from helper T cells, the B cells will differentiate into plasma B cells that can produce antibodies against a specific antigen. And the humoral immune system deals with antigens from pathogens that are freely circulating or outside the infected cells. Okay, so pathogens outside the cells. And then the antibodies produced by the B cells bind to antigens, neutralizing them or causing lysis, so the dissolution or destruction of cells, or phagocytosis. So this slide looks at the different types of antibodies in more detail. So antibodies can be divided into 5 classes: IgM, IgG, IgA, IgD, and IgE, based on their physiochemical, structural, and immunological properties. And it's IgGs which make up about 80% of all antibodies. And IgGs are secreted by plasma cells into the blood. So after an adaptive defense is produced against a pathogen, usually the plasma cells first secrete IgM into the blood. B cell receptors on naive B cells are of the IgM class and occasionally IgD class. IgM molecules make up approximately 10% of all antibodies. So prior to antibody secretion, the plasma cells assemble IgM molecules into pentamers. So that's 5 individual antibodies, and these are linked by a joining J chain. Okay, we can see this on the bottom left hand of the slide. And this pentamer arrangement of the IgM molecules, this arrangement means that they can actually bind 10 identical antigens. However, IgM molecules released earlier in the adaptive immune response do not bind to antigens as stably as IgGs, which are one of the possible types of antibodies secreted in large quantities upon re-exposure to the same pathogen. So IgAs, we're going to talk about in more detail later on, because these are involved in mucosal immunity. Okay, these IgAs, these are mainly found in saliva, tears, breast milk, and mucus secretions of the gastrointestinal, respiratory, and genitourinary tracts. Okay, and these body fluids coat and protect the extensive mucosa. Okay, so that's why the IgAs are involved in mucosal immunity. And similar to the IgM antibodies, the IgAs are secreted as polymeric structures linked with the J chain. However, IgAs are secreted as mostly as dimeric molecules, so that's two antibodies linked together rather than pentamers. So 2 antibodies linked together rather than the 5. Okay, so you can see the secretory IgAs at the bottom right hand of the slide. And then we've got IgEs, which are present in the serum in small quantities, and they are best characterized in their role as allergy mediators. Then we have the IgD in small quantities in the serum. Similar to IgM, B cell receptors of the IgD class are found on the surface of naive B cells. This class supports antigen recognition and maturation of B cells to plasma cells. Okay, so we've talked about humoral immunity, and now we're going to talk about cellular immunity. Okay, again, you'll cover this in more detail in the spring term, but briefly, cellular immunity occurs inside infected cells and is mediated by T cells or T lymphocytes. The pathogen's antigens are expressed on the cell surface or on an antigen-presenting cell. Helper T cells release cytokines that help activated T cells bind to the infected cell's antigen complex and differentiate the T cell into a cytotoxic T cell. The infected cell then undergoes lysis. And now it's important to talk a little bit about immunological memory. So this is the ability of the immune system to quickly and specifically recognize an antigen that the body has already encountered and initiate an immune response. And vaccines produce this immunological memory because of the long-lived memory B and T cells that are produced that can recognize the virus, and these patrol the body for months or years, providing immunity. So a vaccine is a biological preparation to provide acquired immunity against specific diseases. And the immunity to viral infections usually depends on the development of an immune response to antigens on the virus surface and antigens on the virus infected cell. In most cases, response to internal proteins has little effect on the humoral immunity to infection. A successful vaccine works by blocking uptake of the virus upon entry. Vaccines must induce antibodies that bind to the receptor binding proteins on viruses. They are called neutralizing antibodies. So this slide looks at how antigens are selected for vaccines using the flu virus as an example. So the flu virus has two surface proteins, hemagglutinin and neuraminidase, which play essential roles during virus entry into host cells and release from those cells. So the neuraminidase cleaves sialic acid and is involved in the release of virus from the cell, whereas the hemagglutinin binds to cell receptors and attaches the virus to the cell. And therefore, the hemagglutinin is the major target for neutralizing antibodies. So this is why this antigen was selected for the vaccine. So this slide looks at the different types of vaccines that are available. So if we start with whole pathogen vaccines, so as the name suggests, these are vaccines which incorporate the entire pathogen, not just a component of the pathogen. And you can get attenuated vaccines or killed/inactivated vaccines. So if we start with the attenuated vaccines, first of all, so these are live vaccines, and these live attenuated vaccines are derived from disease-causing pathogens, so virus or bacteria, that have been weakened under laboratory conditions. Okay, so they've been weakened in the laboratory, and they'll grow in the vaccinated individual, but because they're weak, they will cause no or very mild disease. And these attenuated vaccines stimulate an excellent immune response that's nearly as good as an infection with the wild type pathogen. There are some disadvantages with this type of vaccine though, including the challenges of the long-term storage and transport of the vaccines, as well as there's potential for the patient to develop some signs and symptoms of the disease during the active infection, particularly in immunocompromised patients. And there's also a risk of the attenuated pathogen reverting back to full virulence. And we're going to talk about that later when we talk about the polio vaccine. So examples of the live attenuated vaccines are chickenpox, German measles, mumps, TB, typhoid fever, and yellow fever. And then the second type of the whole pathogen vaccine is the killed/inactivated vaccine. So these inactivated vaccines are made from viruses, bacteria that have been killed through physical or chemical processes, and these killed organisms cannot cause disease. So the disadvantage of this inactivated vaccine is that inactivated vaccines do not produce an active infection, and so the immune response is a lot weaker than that provoked by a live attenuated vaccine. So typically the response involves only the humoral immunity, so that's the antibody response, and the pathogen cannot be transmitted to other individuals. In addition, inactivated vaccines usually require higher doses and multiple boosters, possibly causing inflammatory reactions at the site of injection. However, on the positive side, these inactivated vaccines do have the advantage of long-term storage stability and ease of transport, and there's no risk of causing severe active infections. Examples of inactivated vaccines are cholera, hepatitis A, influenza, plague, and rabies. So now if we look at subunit vaccines, so in contrast to the whole pathogen vaccines, the subunit vaccines only expose the patient to the key antigens of the pathogen, not the whole cells or viruses. And these subunit vaccines can be produced in two main ways. The first way is by chemically degrading the pathogen and isolating, purifying its key antigens, or the antigens can be produced through genetic engineering. So these are recombinant vaccines. And the advantage of these vaccines are because they only contain the essential antigens of the pathogen, the risk of side effects is relatively low. And examples of the subunit vaccines are anthrax, hepatitis B, influenza, meningitis, papillomavirus, whooping cough as well. And we're going to talk about the hepatitis B recombinant vaccine later on. And the next type of vaccine we're going to talk about are the toxoid vaccines. So these are inactivated bacterial toxins. So like the subunit vaccines, the toxoid vaccines don't incorporate a whole pathogen, they just contain the inactivated bacterial toxins. And these are used to prevent diseases in which the bacterial toxins play an important role in the disease. And the way that these vaccines work is they activate the humoral, the antibody immunity that neutralizes the toxins. And good examples of these, which we've covered before, are botulism, diphtheria, and tetanus. And then we have conjugate vaccines, and these are a specific type of subunit vaccine that consists of a protein conjugated or joined to a capsule polysaccharide. And these conjugate vaccines, they've been developed to enhance the efficacy of subunit vaccines against pathogens that have these protective polysaccharide capsules. And these capsules help them avoid phagocytosis, causing more invasive infection. So these subunit vaccines introduce capsular polysaccharide antigens that result in the production of antibodies that can opsonize the capsule and thus combat the infection. So this slide shows the newer types of vaccines that are becoming available. So these are nucleic acid vaccines. So first of all, we have DNA vaccines, and we're going to cover these in a bit more detail later on. But basically, these are produced by incorporating genes for antigens into a recombinant plasmid vaccine. And then you have RNA vaccines, and these use a piece of mRNA, which will produce some of the antigenic proteins of the disease-causing virus. And then you have the viral vector-based vaccines. And these use low pathogenic, so mainly harmless viruses, and alter them in what you call into viral vectors that will produce some of the same proteins as the disease-causing virus. So to put it simply, these vector vaccines use a weaker virus to transport pieces of the pathogen in order to stimulate an immune response. And there are two different types of these viral vector vaccines, so replicating and non-replicating. And the only difference is the non-replicating viral vector vaccines can't replicate inside the body, as the key viral replication genes have been deleted from the low pathogenic vector virus. And I basically highly recommend you look at the link below, and this is a very good infographic showing all these different types of vaccines. It explains them quite well with diagrams, and it also tells you about how these types of vaccines are being used in the COVID vaccine development as well. So this slide just gives a good overview of the different types of vaccines in picture form. And this table shows you some examples of vaccines in the different categories, but this isn't an exhaustive list of the current vaccines. Okay, there are many more of these. This is just some examples.